Hi, everyone. The video you're about to watch is part of the Age Presents lecture series. The series is produced by the American Aging Association to provide an introduction to foundational and advanced topics in geroscience. As you watch this video, I would like for you to keep 3 points in mind that will help you understand the importance of biological aging. 1, aging is the primary risk factor for disease, including cancer, metabolic syndrome, and most neurodegenerative diseases. 2, the scientific approaches that are being used here today, such as the model systems that help researchers learn more about the mechanisms within these age-related diseases. And 3, mostly centers around you. What is your interest in aging research? Whether it's biological, translational, or clinical, how would you like to contribute to the field? And if you're not already a member of the American Aging Association, I would encourage you to visit our website to learn more about us and consider joining today. AGE members receive free access to the journal Geroscience, reduced registration for our annual meeting, opportunities to interact with many of the leading researchers in the field, and access to all of the lecture slides and the notes that are being used here today. And there's more. We have a very active trainee chapter with many opportunities for networking and career development. And last but not least, I would like to acknowledge and say thank you to our sponsors for the Age Present lectures. The University of Washington Healthy Aging and Longevity Research Institute, the Jackson Laboratory Aging Center, and the Nathan Schott Centers of Excellence at both of these institutions. Thanks for listening and enjoy the lecture.
Hello, my name is Nausica Arnoult. I'm an assistant professor of molecular, cellular, and developmental biology at the University of Colorado Boulder. My lab works on telomeres and DNA repair and how they function in aging and cancer. So today I'm going to talk about the function of telomeres during aging, both cellular aging and the consequences for the aging of our body. So as you all know, we all age, and the aging of our bodies is not completely well understood, and there's plenty of factors that participate in organismal aging. But for sure, it's linked to the aging of our cells. And this aging, there's plenty of factors that participate in cellular aging, but one factor is controlled. It is the number of divisions that a cell can do. And that's controlled by telomeres. So that's what I'm going to explain today. So this was discovered in the 60s by Len Hayflick, that when you put cells in culture in a specific environment, medium, that is like a feeding for the cells, that allow them to divide, you can do that for a certain number of time, but after a certain number of divisions, the cells stop dividing. So they're still alive, they're still there, but they will not grow anymore. So this is what you see here. These are young cells. The brown thing is the nucleus, so it's the part of the cells that contains the DNA, and you see they are very elongated cells. And when they become old and stop dividing, they're much bigger, they change shape, they change, a lot of things are changing in these cells. So they do not die, but they don't divide anymore, and they completely change morphology and physiology. These cells are called senescent. So how Len Hayflick discovered that is quite interesting, because people were culturing human cells since some time. And to culture human cells and have all the factors that you need to make them grow, the easiest is to take the serum of blood. But when people were doing that, it was not very clean, and they were actually introducing new cells in their medium. And what Len Hayflick did was to make cell culture much cleaner, filter the serum. So actually the cells that he did put in culture at the beginning were the same as the one he had in the end. And so that's how, for the first time, it was found that human cells cannot divide, have a limited potential of division. And that limited potential of divisions is called the Hayflick limit. So how that worked was not known at all at the time, and it took a good 30, 35 more years to discover that this was linked to telomeres. So that's what I'm going to explain during this lecture. But first we need to understand what are telomeres. So telomeres are the ends of our chromosomes. So you see here, that's a photo of metaphase spreads, which is when chromosome condense between during mitosis. And so you see nicely the chromosomes in blue. So the blue is basically the DNA. And there's probes that target telomeres in green and red. And you see that these telomeres are really the portions of telomeres that are located at the end. And their function is to protect chromosome ends. So if I want to make an analogy, one of the classical analogy made in the field is that telomeres are like the little tips that protect laces. So if chromosomes are the laces, then telomeres are this little tip at the end. And if your tip gets damaged, then your laces start to fall apart. So these tips are very important. Now, let's see what telomeres exactly do to protect chromosomes, not exactly like laces. So they have several functions, and all these functions have evolved because chromosomes at the beginning, I mean in bacteria, the first forms of life, the genomes were round, so there was no end. And as species evolved and became more complex, there was a need for bigger genome, and that could not work with a circular genome. So this genome fragmented in several chromosomes, and these chromosomes have ends. And that posed several problems that need to be addressed. And this is addressed by telomeres. So the first problem is the end replication problem. So telomeres solve the end replication problem, and we're going to see why. How? So what is replication first? When you have a cellular division, you basically do two cells out of one cell. And to do that, you need to duplicate the entire content of the cell, including the DNA. And so the whole copy of the DNA to make from one genome, two genomes, so each cell can have their own genome. This process is called replication. It's really the name, the scientific name given to duplication of DNA. So if we want to take another analogy here, I would say that your genome is basically a manual that contains all the explanation of the different tasks that every cell in your body needs to accomplish. So imagine it's like, a manual, a book, and doing the replication is like copying the entire book. So that in the end, each daughter cell has its own manual and can figure out how all the tasks in the cells can be achieved. There's a little problem though, is the copier works really well on circular genomes, but it doesn't work very well on linear genomes. So the problem is it cannot copy the very beginning and the very end. It's a little too complicated to explain why, but really each time you copy the chromosomes, you're going to lose the very last pages. And at some point, you lose too many pages that the cells would not be able to figure out the manual. You imagine you have a manual to build something and you don't have the first or the last pages. That is problematic. So there's basically two solutions for that. Either you fix the copier so it can copy the manual entirely each time, but technically it was not possible. So the cells adopted the other solution, which is simply to add blank pages at the very beginning and at the very end of the manual. So as the cells divide and the copier doesn't copy the very ends, well, what happens is you lose blank pages, but you retain all the information of your manual. And in terms of biology, these blank pages correspond to non-informative sequences, and these non-informative sequences are telomeres. So in the case of telomeres, these sequences of DNA that do not carry out any information are repetitive sequence. It's TTA, GGG, TTA, GGG out of thousands of times. Telomeres actually means, so telo is for the Greek, that means the end, and mere is the part, segment. So it's a repeated segment at the end of chromosomes that do not carry any information. So as cells divide, instead of losing genes and sequences that are important for the cell, you basically shorten that repeat of TTA GGGs. And so that's how telomeres solve the end replication problem. But there's another problem that came from the fact that chromosomes are linear. And that's the end protection problem. And to explain the end protection problem, I need to take a little detour and explain what is DNA repair. So our DNA gets broken and damaged constantly. And therefore, the cell has evolved several ways to sense and repair the breaks. So this break can happen just naturally due to endogenous stress or to external factors such as cosmic radiations or genotoxic chemicals like cigarette smoke, for example. So how does DNA break sensing and repair works? So the first thing is that there's some proteins that are going to see that the DNA is broken, so that's the sensing, and it's going to send a signal to the cell to stop dividing, and then it's going to assess if there's few or too many breaks. If there's few breaks, the cells can repair it. So in that case, it recruits more proteins that are going to repair the break. If there's too many breaks, it's just too dangerous for the cells to repair it because there's too many chances that mistakes are going to be made. And in that case, the cell will send a signal that will lead to permanent cellular arrest, senescence, as we've talked before, or to death. So why do I talk about that, and what is the relation with telomeres? If you look at how DNA break looks like, in this case, DNA double-strand breaks, where both strands of DNA are broken, and you look at telomeres, they basically look exactly the same. And so if left just like that, there's no way for the cell to know that a chromosome end is not broken DNA and that doesn't need to be repaired and it doesn't need to be sensed as damaged. Which would be a big problem because there's an end at every chromosome. So if each chromosome end was sensed as DNA break, that reaches far the limit of the number of breaks that the cell can sustain, and therefore that would directly send the cells to senescence. So that's what the second function of telomere is really to, at the ends of chromosomes, say, okay, this is not a double-strand break, and prevent the cell to recognize it as double-strand break and repair it and send a damage signal. Okay, now we're going to see how that works. So I've told you that telomeres are TTA GGG repeats, and you know that DNA is double-stranded, so I'm going to represent the second DNA strand that is complementary to TTA GGG. So this double-stranded DNA of telomeric repeat is between 3,000 and 15,000 bases. But at the end of it, and this is very specific to telomeres, there's this small 100, 300 bases of TTA GGG that is single-stranded. And that is called an overhang. And this is really important because this overhang will enable the invasion of the double-stranded DNA to loop back. So that forms a loop. You see here is the single-strand DNA that invades the double-strand DNA. And that enables to form a loop. And that's a beautiful electron microscopy image of a telomere forming this type of loop. That's called a T-loop, for telomeric loop. So you understand from here that if you have this structure, it no longer looks like a double-strand break because it hides the end of the chromosome. And that's one way telomeres inhibit DNA damage signaling, is by hiding the end of chromosomes in a loop structure. The second thing is that TTA GGG repeats recruit a specific protein complex called the shelterin. So the shelterin binds just to this TTA GGG, and they have two functions. The first function is actually to help form this T-loop. And the second function is to directly inhibit DNA repair. And we're going to see later why this is really important. So this is really how telomeres solve the end protection problem by both the T-loop and the shelterin protein. They hide the end of chromosomes and they inhibit the proteins that do DNA damage sensing and DNA repair. So what we've seen so far, the end replication problem and the end protection problem, leads to the third function of telomeres, which is linked to aging, the mitotic clock. So mitosis is the moment when cells divide, split into two cells. So the mitotic clock is the capacities of cells to count how many divisions they've been through. And that uses both, as I told you, the end replication and the end protection problem. Okay, so how does that work? Here is a graph of how telomeres shorten. You remember, because of the end replication problem, telomeres shorten at each cycle of division, at each replication. And that's one cell. And each of these blue dots that you see represents a telomere. At this point, the cell is very young. So all telomeres are very long. They form the T-loop and they have plenty of shelterin protein on them. As the cell divides, the telomeres start shortening. This process is heterogeneous. Not all telomeres are going to lose exactly the same number of telomeric repeats. And so that's why there's quite some heterogeneity in the telomere length as the cells divide. So they start shorten, but at this point they're still long and fully protected. But as they keep shortening, they're going to have more and more short telomeres. And basically, when the telomeres are shorter, it's getting more and more difficult to form the T-loop. They still can form the T-loop, but the probability of having one telomere fail to form the T-loop is increasing. And so at some point, one telomere will fail to form the T-loop, so it still has sheltering protein on it, but it opens. And this deprotected telomere will be recognized as a double-strand break. At this point, though, it elicits just a basal DNA damage response that is not going to lead to cellular arrest. So the cell can still overcome this problem. But as cells keep dividing, at some point it has more and more short telomeres, and the number of telomeres that fail forming the T-loop is going to reach a point that the cell cannot accept anymore. So this has been established as 5 deprotected telomeres. So 5 telomeres that can't form the T-loop makes that there's enough DNA damage for the cells, enough telomeres that are seen as double-strand breaks. And that will send the cells to senescence, so permanent cell cycle arrest. It's important to note that at this point, the telomeres are still long enough that they have still sheltering proteins. So they still inhibit DNA repair. And so if telomeres repair, they're going to fuse. So there's going to be chromosome fusions, and these chromosome fusions lead to genomic instability. And so it's very important that when cells enter senescence, they don't do telomere chromosome fusions because of the sheltering protein, so their genome is still very stable. So anyway, this is how, by shortening and at some point being deprotected and not being capable of solving the end protection problem any longer, this is how telomeres enable cells to count how many divisions it went through and stop the cells when it reached too many divisions. So that's really the process, and that's what explains the half-life limit. As you take young cells with long telomeres, they can divide, but at some point, for each cell, you have too many telomeres that are too short, and the cells enter in senescence. Okay, and this actually participates in the aging of our body, because as we age, more and more of our cells have reached this half-life limit, and the senescent cells are accumulating in our body, and they participate in the aging of our tissues. Okay, so do all cells have a mitotic clock? And that's an interesting question. So imagine they do. Imagine every single cell in your body have their telomeres that shorten. Then what happens is, as an adult, you already have telomeres that have shortened a little bit. So when you have children, these babies will be made out of these shorter telomeres and be born with telomeres that are as short as their parents. And these 10 babies will still have their telomeres shorten, and as adults will have babies with even shorter telomeres. And you can imagine that after a few generations, that's basically the extinction of the species. So that cannot work, obviously. And that's why the clock is reset in germline. So babies always start with brand new telomeres. So they're a brand new clock. And how does that work? So that works by having a specific enzyme, so a specific protein in our cells that is called telomerase, that has an RNA that is a telomeric sequence, and this telomerase is going to use this RNA to as a template to elongate telomeres. It's a very unique enzyme in our body that can make DNA out of its own RNA template. So it's going to elongate telomeres, and that enables cells to replenish telomeres as they divide. So if this is telomere length and this is division, and if you look at germline or embryonic stem cells, as the cells divide, the telomerase is going to replenish the telomeric sequence, and it will not shorten. So these cells can grow and grow and never have telomere shortening. But as the cells then differentiate, so if you go from embryonic stem cells to multipotent stem cells, they start repressing the expression of telomerase. So there's going to be less telomerase enzymes in the cells, and so then it's not going to be capable of replenishing all the telomeres that shorten. So in multipotent stem cells, there's going to be some kind of telomere shortening as the cells divide. And now this happens as the cells keep differentiating, and in cells that are completely differentiated, telomerase is completely repressed. So there is no expression at all, and in these cells, well, the mitotic lock happens. Okay, so if there's a way to avoid the mitotic lock, why do we even have one? What's the point? So let's go back to DNA repair. I've told you that DNA breaks constantly and gets repaired. The problem is this repair is often imperfect, and it leaves scars in the DNA, which can be mutation, deletions, translocations, so a lot of different things that alter the DNA sequence. And the problem is that these mutations can lead to cancer. So basically, the mitotic clock is going to say, okay, so as cells divide, they accumulate more and more of these mutations, and are more and more likely to develop cancer if you let them divide too long. So that's why it's important for cells to say, okay, after a certain number of divisions, it's better to stop, because the chances of making a cancer become too big. So if we have this mitotic clock, how can cancer even develop and in these differentiated cells? How does it work? So let's go back to our telomere shortening graph, and you see here the cells are going to enter senescence, which is really the first anti-tumor barrier. This senescence works by a signaling that is mediated by different proteins, especially p53 and RB. The problem is, at this point, cells have already accumulated mutations. So most of the time, these mutations are not a big deal. Either they happen in non-coding sequence, or, it's not going to make too much of an effect on the cells. But sometimes, by chance, this mutation will occur in these two genes. And then if you don't have p53 and you don't have RB, you lose the signaling, you lose the tool to tell the cells to stop dividing. And if you lose that, well, then the cells will keep dividing and they will keep having their telomeres shortening. And so that's what happens in these few cells that have, we say, bypassed senescence. So PV3 and RB are really like checkpoints in the cells, and it's really like removing the checkpoint. So you remove the checkpoints, the cells keep dividing, and the telomeres keep shortening up to a point where, so they accumulate way more of this, but now the cells cannot care anymore because there's no checkpoint, so there's no way to arrest the cells that accumulate that. But now they're going to have so short telomeres that they can't retain sheltering proteins anymore. And if they can't retain sheltering proteins, then they can't inhibit DNA repair. And they are going to repair like double-strand breaks too. So if your DNA breaks, most of the time the repair will be by just bringing the two ends and like gluing them together. Well, the cell is going to do exactly like that with telomeres, except this time it's going to take two chromosomes and glue them together, fuse them together. And that's called a chromosome fusion. The problem with chromosome fusions is that when the cells will go to the next division, usually what it does is take chromosome and separate it in two parts. So one part here is going to go in one cell, and one part here is going to go in the other cell. But when you have two centromeres, so two regions that bring the chromosomes together, because two chromosomes fused, this is very hard to do. And this will create tensions and very often this fused chromosome will break and generate more fusions, more instability. And that basically leads to a cycle of genomic instability that in a vast, vast majority of the time will lead to cellular death. So the cells that go beyond senescence, they are going to keep dividing, and at some point they will die. So this is called crisis because of this huge genome instability that occurs in a very short time. And that's the second anti-tumor barrier, because really almost all the cells will die from that. However, during that instability, some cells will acquire a way to elongate their telomeres. And when they do that, they stabilize the fusions, they stabilize the genome, and that stops the crisis process. And now our telomeres are long enough that they are not, well, they're basically stabilized and protected again. And the way cancers do that, there's two ways of doing that, but in the vast majority of the time, in about 90% of cancers, they do that by reactivating telomerase. So that is why telomerase is switched off as cells differentiate, because otherwise it would be tumor prone. So most of the cancers have telomerase expression. The other 10% used alternative mechanisms. But in any case, in all cancers, there is a way to elongate telomeres, and that enables the cells to not have a mitotic clock anymore. So they have an unlimited potential of division, which is really one of the most important characteristics of a cancer, is a cell that just divides out of control. So basically, if we want to summarize from here, telomeres are really going to balance aging and cancers. And what's interesting is, so in human cells, you switch off telomerase. But in other organisms, small organisms that don't live long, like mice or plenty of small organisms, there is no switching off of telomerase, because these species, they basically die before they have time to develop cancer, that are getting eaten by some other animal in nature. And so during evolution, there was basically a a moment when an organism, a species, lives long enough, it is important to have a mechanism to prevent cancer. And this mechanism is to force cells to age, to stop them dividing. The other mechanism is actually to have a very good DNA repair system. And in species that live very, very long, there's more checkpoints. So, for example, more copies of p53. And there's a very good, very strong mechanism of DNA repair, which prevents this accumulation of mutations. But it's always a balance during evolution between the cost and the advantage that it brings. So now you see how telomeres really control the mitotic clock and therefore control how cells are aging. Now I wanted to take a few minutes to talk about the aging diseases that are associated with dysfunctional telomeres, because of course this requires a lot of proteins, so there are plenty of genes that can be mutated and lead to disease. So this is how normal, it's a representation of how normal telomeres shorten. You start as a baby with long telomeres and they shorten slowly as you age. But some individuals are born with very short telomeres, abnormally short telomeres, and so their clock will not be set like a normal clock. So they already have all the telomeres at birth, and so as these cells divide, they will reach a critical point way earlier, prematurely. And most of these short telomeres at birth syndromes are due to a defective telomerase. So not necessarily the enzyme, so it can be the enzyme of telomerase, it can be the RNA, or it can be there's actually some proteins that bind to the RNA to stabilize it, so it can be these proteins. So there are several syndromes of premature aging that are due to a defective telomerase. And so in this case, as the embryo develops, these germline cells cannot fully replenish their telomeric sequence, and the baby that is born out of that process already has very short telomere at birth. A second problem is when individuals are born with normal telomeres, but they shorten way too fast. And that happens when there's a problem during telomere replication. So I've told you that there's this copier that copies all the genome. And telomeres are a little bit specific sequences that are not necessarily easy to replicate. And they need specific proteins to help. And if these proteins are defective, then the copier itself is defective. So that means that at every replication, instead of losing 100 base, some telomeres will shorten rapidly. And so these individuals with this type of syndromes are born with, they start aging completely normally. They don't have any symptoms at birth or during childhood. But as they reach a certain age during adulthood, they start having really this accelerated aging. And then the last problem is defective telomere protection. So these are individuals that are born with normal length telomeres, but even these normal length telomeres are recognized by the cell as damage. And so they trigger this damage signaling response that leads cells to go to senescence way before they should. And that is usually a defect in forming the T-loop for different reasons, for different proteins that, so it could be different proteins that help forming that T-loop. But in the end, you have these long telomeres that can't form a T-loop and that are recognized as if they were short, and that lead cells to go to senescence even though they are young and healthy otherwise. Okay, so these are all very dramatic syndromes, but if you go back to healthy individuals with normal telomeres, while unfortunately, we're not all equals in terms of telomere length. So we all start with long telomeres, but long doesn't mean exactly the same for all of us. And we basically inherit telomere length from our parents. And so some individuals are born with very long telomeres, while others are born with shorter telomeres. So that's a graph that represents basically the distribution of telomere length as we age. And you see that there's a big difference between individuals, with people born with extremely long telomeres and some with way shorter telomeres. So there's not much we can do about that. But what we can do, on the other hand, is try to have an effect on the way telomeres shorten. So there's been many studies, including by Elizabeth Blackburn, who won the Nobel Prize for the discovery of telomerase. And she has shown that a lot of factors influence how fast telomeres are shortening. So for example, excessive stress or unhealthy behavior like no exercise, bad food habits, all this participates in accelerating the shortening of our telomeres. So we all know we should live a healthy life with, healthy food habits, exercising, controlling stress. We all know that, but you should also know that these healthy habits are very good for your telomeres, and that's one of the many ways that these healthy habits keep people healthy and keep people from aging. So take care of you, but also take care of your telomeres. And with that, I just have a few references if you want to read a little bit more about telomeres and telomeres and aging. So some reference about telomere structure, sheltering, and telomere protection. Some references about the telomere-associated aging diseases, and some about senescence and aging. And with that, I want to thank you and